We've all stood there, choosing our cannabis based on those familiar labels. Indica, sativa, hybrid. You kind of expect a certain effect, right?
Yeah, definitely. Relaxing, energizing. We've built these expectations.
But what if those trusted names don't actually tell you the real chemical story of what's inside?
Mm-hmm.
Today, on the deep dive, we're unpacking a really groundbreaking research paper. It meticulously analyzed the phytochemical diversity of commercial cannabis across the United States.
It's a big one.
And this isn't just about cannabis, really. It's a powerful case study for any complex botanical product. Huge implications for consumer safety, marketing, even future medicine.
It's a very common experience for consumers, isn't it? We
Right.
rely on these labels, often without, well, truly understanding the science behind the plant itself. This paper really pulls back the curtain.
And it does so on an incredible scale. Can you walk us through the sheer ambition of this research? What kind of data are we talking about here?
Oh, it's massive, just massive. The researchers looked at almost 90,000 samples of commercial cannabis flower.
90,000. Wow.
Yeah, not just a few hundred. And these came from 6 different U.S. states.
Oh,
California, Washington, Oregon, Michigan, Florida, Alaska. So a pretty wide net.
okay.
And crucially, they went beyond just, THC and CBD.
Mm-hmm.
They analyzed both the full spectrum of cannabinoids and the terpenes.
Ah, the terpenes, the smell compounds.
Exactly, those aromatic compounds. They looked at the whole chemical picture.
So with all that data, what was the most significant, maybe even surprising finding this paper uncovered, especially about those labels we all trust?
Well, okay. If you've ever picked out cannabis based on whether it's indica or sativa, you might need to sit down for this.
Uh-oh.
Because what they found fundamentally challenges how we've always understood these products. Basically, those common labels, indica, sativa, hybrid.
Mm.
They simply do not align reliably with the actual chemical makeup of the plant.
Really? No alignment.
Very little,
Yeah.
statistically speaking. It's a huge disconnect between, the marketing perception and chemical reality.
Okay, let's unpack this. Let's start with the building blocks. Cannabinoids. We hear about THC and CBD all the time, but what are their original forms in the plant before you light up?
Right. So in the plant itself, you mostly find THCA and CBDA. That A stands for acid.
Acidic forms, okay.
Exactly. They're the raw, non-psychoactive precursors. Think of them like ingredients before you cook them. Heat, like from smoking, vaping, or cooking, is what activates them. It converts THCA to THC and CBDA to CBD.
And that's when they interact with our body's endocannabinoid system, giving us those potential effects.
Precisely. That's when they become pharmacologically active in the way most people recognize.
Now, THC and CBD get all the headlines, but I remember reading there are dozens of other cannabinoids present. Are these just sort of background noise, or do they matter?
Oh, they definitely matter, or at least they could matter a lot. While THC and CBD are the most abundant, sure, there are dozens of others. Things like CBGA, CBCA, CBN, THCVA, CBG, THCV.
Wow, alphabet soup?
Huh? Yeah, a bit. But even in small quantities, these minor cannabinoids are incredibly interesting. They show a whole range of pharmacological properties, potential medicinal value that's still largely unexplored.
So they're not just noise.
Not at all.
Yeah.
They're like the supporting cast that might have starring roles we just haven't figured out yet.
Okay, so with thousands of samples, how did the scientists categorize them based on cannabinoids? Did they find distinct groups?
They did. They classified samples into 3 main chemotypes based on their THC to CBD ratios.
Chemotypes, chemical types, basically.
Exactly. And the results were pretty stark. The overwhelming majority, a staggering 96.5%, were THC dominant.
96.5%.
Yeah. Then you had CBD dominant at just 1.4% and balanced THC CBD making up the rest, about 2.2%.
So the market is almost entirely tilted towards high THC.
Overwhelmingly so. It really shows that most of the cannabinoid variation you actually find in commercial cannabis comes down to the levels of THC, CBD, and also CBG, another cannabinoid.
Interesting. Now, beyond cannabinoids, you mentioned terpenes. We know they affect the smell, but what's their deeper significance in this study? Are they just about aroma?
Not just aroma, although that's important for consumer experience. Terpenes are secondary metabolites, yes, responsible for those distinct smells, piney, citrusy, earthy, whatever.
Right.
But beyond smell, they're also pharmacologically active themselves. And the paper suggests they might actually be very reliable markers for classification.
Right.
Maybe even more reliable than indica sativa.
More reliable than the labels we use now.
That's what the data points towards. The most prominent terpenes they found were myrcene, beta-caryophyllene, and limon.
Okay.
And on average, total terpene content was about 2% by weight. Interestingly, it correlated positively with total cannabinoid content.
So more cannabinoids often means more terpenes.
Seems like it. It suggests a deeper botanical connection, maybe shared pathways in the plant.
And did they find patterns in which terpenes show up together? Like, do certain smells tend to cluster?
They did, and it's quite telling about the plant's biochemistry. They found robust positive correlations between certain pairs, like alpha and beta pine. Those piney smells often appear together.
Makes sense.
And beta caryophyllene, which is kind of peppery, often shows up with humulene, which is more earthy or hoppy. This isn't random. It likely reflects the underlying biochemical synthesis pathways. The plant sort of has preferred chemical recipes.
Fascinating. Okay, now for the part that might ruffle some feathers, the myth of indica versus sativa.
The big one.
For years, this has been the bedrock of how consumers classify cannabis. Just how deeply entrenched are these folk theories? Relaxing indica, energizing sativa.
Oh, they're absolute central. It's the primary language of the market. You
Oh.
know, whether you're in a dispensary or talking to friends, indica means sleepy or body-focused. Sativa means uplifting, creative, head-focused. Hybrids are somewhere in the middle. It's how almost everyone chooses.
And what did the chemical evidence, looking at those nearly 90,000 samples, reveal about these deeply held beliefs?
Well, a paper delivers a pretty stark reality check.
Uh-huh.
The analysis showed that indica, hybrid, and sativa labels do not reliably distinguish samples based on their actual chemical content.
Not reliably, so sometimes they do.
Statistically, no.
Mm.
When they mapped out all the chemical profiles using things like PCA plots, what they saw was basically a jumble. The samples labeled indica, sativa, hybrid were all highly intermingled.
So an indica could look chemically identical to a sativa.
Pretty much indistinguishable, yes, based purely on the measured cannabinoids and terpenes. It's like buying fruit labeled apple, but sometimes getting a pear. Chemically, the label doesn't guarantee the contents.
So the marketing, based on these labels, the paper calls it specious. That's a strong word. What does this massive disconnect mean for us, for users, and how strains are marketed?
It fundamentally undermines how consumers currently make choices, doesn't it? If you're buying a product expecting a specific effect based on its label, and that label isn't actually backed by the chemistry, well, you're essentially buying blind.
Right. It leads to unpredictable experiences?
Exactly.
Yeah.
And it creates a significant challenge for consumer trust.
Yeah.
How can you rely on a product if the label doesn't mean what you think it means?
That's a huge paradigm shift. Okay, so if the traditional labels aren't cutting it, the study doesn't just point out the problem, right? It proposes a solution.
It does. That's the constructive part. The authors propose moving towards a classification system based on observable chemotypes.
Chemotypes again, based on the actual chemistry.
Precisely, using terpenes and cannabinoids. This moves us away from these vague folk categories towards precise chemical profiles that you can actually measure and verify. Science-based, essentially.
Okay, so how did they define these new clusters? When they grouped the THC-dominant samples, what kind of chemical signatures emerged?
They used a statistical method called k-mean clustering. For the THC-dominant samples, which was most of them, they found 3 major terpene-based chemotypes.
3 main groups based on terpenes.
Yeah. Cluster I was characterized by high caryophyllene limon, think spicy, peppery, often citrusy notes.
Okay, I can almost smell that.
All right. Then cluster 2 was defined by high marcine pine. That leans more towards earthy, piney, maybe more traditionally indica-like aromas, though we have to be careful with that link now.
Gotcha. And cluster 3?
Cluster 3 was interesting. It was characterized by high terpinolene myrcene. Terpinolene is less common, often described as floral, piney, maybe a bit herbal. And notably, this cluster also had somewhat higher levels of CBG.
Higher CBG.
Yeah, hinting at potentially unique effect profiles that aren't captured by just THC, CBD, or the old labels. This system moves us towards understanding why a strain might smell or feel a certain way based on its dominant chemicals.
So instead of a guess based on a folk name, this offers a chemical map. What are the practical benefits here? This sounds potentially revolutionary for consumers and maybe medicine.
The benefits could be huge. First, it's demonstrably better at actually discriminating between samples than the old indica sativa labels. It reflects real chemical differences.
Which means more predictability.
Yes, exactly. The paper says it provides new possibilities for systematically categorizing commercial cannabis based on chemistry. And this could profoundly improve consumer safety and clinical reliability. Imagine knowing the actual chemical profile ensures consistency and more predictable effects. It's about bringing scientific rigor.
Yeah, that consistency is key, especially for medical use.
Absolutely.
And it makes me think, This sounds like a blueprint not just for cannabis, but potentially for lots of other plant-based products too, right? Could this chemotype approach be extended?
I think so, yes. This approach has much wider potential applications. Think about herbal supplements, essential oils, industries where consumers also face a lack of standardized chemical information.
True, you often just get a plant name.
Right. Applying chemotype classification there could bring similar benefits, quality control, consistency, maybe more predictable effects. It's a framework for understanding complex botanicals better.
Okay, let's switch gears slightly to another big concept in cannabis, the entourage effect.
Ah, yes, the entourage effect.
Yeah.
Always comes up.
The idea that all the compounds, cannabinoids, terpenes, maybe others work together synergistically, creating effects different from just isolated THC or CBD. Is this scientific fact or more of compelling theory at this point?
It's a very compelling hypothesis, rooted in how complex plant medicines often work. But the paper is careful here. It highlights a crucial tension. There's currently limited suggestive evidence for it, in cannabis specifically.
Limited evidence, despite all the buzz.
Yeah. Whether distinct ratios of compounds consistently yield different subjective or therapeutic outcomes in humans is, quote, unknown and still a topic of debate. The anecdotal evidence from users is strong, people feel differences, but hard, controlled scientific proof. That's still largely elusive.
Okay, so why is it so difficult to study this properly? Why can't scientists just test different combinations and see what happens?
Well, the paper points directly to onerous regulations. Conducting human studies, in vivo studies with cannabis, is already challenging.
Right, because it's still federally illegal in the U.S.
Exactly. Now imagine trying to get approval, not just for THC or CBD, but for complex mixtures of dozens of compounds in precise ratios, administered in a controlled way. It's a regulatory and logistical nightmare, frankly. Very expensive, very difficult to get approved and execute well.
That makes perfect sense. It's a practical barrier. So what kind of research is needed then to move this from hypothesis to potentially proven science?
Well, the paper is clear. Carefully controlled in vivo studies are needed. We need human trials.
Okay.
And crucially, future clinical studies should test formulations that actually mimic the cannabinoid and terpene ratios consumers encounter in real products, not just isolated compounds or weird ratios nobody actually uses.
Test what people are actually using.
Right. And this raises a fascinating question. If the entourage effect is proven to be significant, how might that shift drug design or therapy in the future? Would we move away from single-molecule drugs towards complex, multi-compound botanical formulations? It could really change things.
Revolutionize plant-based medicine, potentially.
Could do, yeah.
Okay, shifting gears again, let's talk policy, because here's where things get really tangled. The existing U.S. legal definitions, especially that 0.3% THC limit for hemp. How does that line up with the chemical reality found in this study?
It doesn't line up well at all, especially for CBD products. It's a significant and problematic disconnect for the industry.
How so? What did the data show?
Get this. The study revealed that a staggering 84.5% of CBD-dominant samples had total THC levels above 0.3%.
84%. So most CBD flower wouldn't legally be hemp.
By the strict letter of the U.S. federal law defining hemp based on total THC, including THCA.
Yes.
A substantial fraction of what's grown and sold is CBD-dominant cannabis. Stuff with very high CBD and low THC ratios technically fails the legal definition of hemp.
Wow, that's a massive hurdle for CBD growers and manufacturers, isn't it? The law seems completely out of step with the plant's biology.
Absolutely. It creates this huge legal gray area or even direct conflict for an industry trying to operate legitimately with non-intoxicating plants. It makes you really question the definition.
Yeah, it raises the question, should THC legality be based on the ratio of THC to CBD rather than just an absolute arbitrary 0.3% threshold, especially if the plant clearly isn't intoxicating?
That's a major policy debate spurred by this kind of data. And thinking bigger, this lack of consistent, scientifically grounded definitions, it could really mess up international trade in cannabis and hemp products down the line. Different countries, different rules. It creates barriers.
A global patchwork of confusing laws.
Exactly, based on outdated or incomplete science.
Okay, beyond the legal mess, let's talk about the plant itself. This chemical variation we see, cannabinoids, terpenes, what drives it? Is it mostly genetics like the plant's DNA, or is it the environment, how it's grown?
It's a classic nature versus nurture question, and the answer is likely both. Phytochemical variation comes from genetic differences between strains, absolutely, but environmental conditions also play a big role. Things like light intensity, soil nutrients, water stress, climate.
So the grower has some influence?
Definitely. However, The study couldn't fully separate these factors for these samples. But they did find consistent chemical patterns, especially for terpenes, across different regions.
Consistent patterns across states.
Yeah, which strongly suggests a powerful genetic component is at play. The plant's underlying DNA seems to be a major driver dictating the potential chemical profile, even if the environment fine-tunes the expression.
And what's really striking from the data is how, well, uneven the current commercial cannabis landscape is chemically illiterate. Did the study highlight any untapped potential there, areas we haven't explored?
It certainly did. I mean, remember, 96.5% of samples were THC dominant.
Right, incredibly skewed.
And of those, a massive 87.4% belonged to just two of the three main terpene clusters they identified, Cluster 1 and Cluster 2.
So we're basically consuming a very narrow slice of the plant's full chemical potential.
Exactly. It's an incredibly narrow selection. This naturally points to a huge area for future work. Breeding new cannabis chemotypes not represented in the current commercial landscape will be a key area of future innovation.
Breeding for different chemical profiles.
Yes, there's enormous potential for academic research and product innovation in the breeding of new varieties with higher levels of other cannabinoids, those minor ones like CBG, THCV, CBDV, and unique terpene combinations. Imagine cannabis bred not just for high THC, but for specific therapeutic effects linked to these other compounds, maybe without strong intoxication.
That's a really forward-looking idea. It makes me wonder, could we use advanced tech here, like CRISPR gene editing or genomic selection to, design specific chemotypes for medical or recreational use? Create highly targeted effects.
The potential is certainly there, theoretically. Genetic editing could, in principle, allow for precise control over which cannabinoids and terpenes the plant produces, and in what amounts.
Super cannabis, tailored to order.
It's a tantalizing prospect for creating highly consistent targeted products. But of course, it also raises important ethical and ecological questions. How do we manage such powerful biotechnologies in agriculture? What are the downstream effects? It's not just about the science, but the societal implications too.
Absolutely. Which brings us neatly to a major point for consumers today, the illusion of consistency in commercial cannabis, especially with strain names.
Ah, the strain names. Blue Dream, OG Kush, Sour Diesel.
Exactly. They're used everywhere to market products, implying distinct, reliable effects. But how consistent are they really, according to this chemical analysis?
That's a critical question for consumer trust, and the findings were mixed, leaning towards inconsistent.
Mixed? How so?
Well, the study found some strain names were more chemically consistent than just random chance. For example, Purple Punch samples were 96% likely to fall into that cluster I chemotype, high caryophyllene limon.
Okay, so some consistency there.
Some. But many other popular names showed significant variability across different cultivators, like Tangie samples only clustered together 62.5% of the time. That means over a third of the time, Tangie from one place might be chemically quite different from Tangie somewhere else.
Just a gamble.
Pretty much. Even the most consistent names still displayed, quote, non-trivial levels of variation. It's like a brand promising the exact same coffee blend every time, but sometimes it tastes noticeably different.
And here's the kicker, right? Because cannabis isn't federally legal in the U.S., the paper states there are no enforced rules for the naming of cannabis varieties. What does that lack of regulation mean for consumers?
It means chaos, essentially. Legacy classification systems and names, often carried over from the illicit market, persist without any real standards or verification. This leads to what the paper calls unwarranted trust in product predictability based on a name.
Unwarranted trust. Ouch.
Yeah. There's no official body ensuring that Blue Dream from grower A is chemically similar to Blue Dream from grower B. This illusion of consistency directly affects consumers, obviously, but also branding and trust within the industry itself.
And for medical users, where consistency is paramount.
It fundamentally undermines medical applications. If you need a reliable effect profile for a condition, this inconsistency is a major problem.
So how can the cannabis industry move past this naming confusion? Are there lessons to be learned from other regulated industries, like, say, food labeling with the FDA or USDA?
Absolutely. The industry could certainly learn a lot from agencies like the FDA or USDA about creating transparent, science-backed naming and labeling standards. Consistency is key in those fields.
And what about technology? Could things like blockchain, better lab certification, maybe even unique barcodes for cultivars help improve trust?
Definitely. Those are promising avenues. Blockchain could provide immutable traceability tracking a product's chemical profile from seed to sale. Robust, standardized lab certification is crucial, and maybe unique cultivar identifiers, like a chemical barcode, could give consumers verified information about the actual chemotype, regardless of the catchy strain name. It's all about building a predictable, reliable, transparent market.
Transparency seems key. Okay, so let's bring it back to the core message about the old labels. To reiterate, the Indica hybrid sativa system does not reliably distinguish samples based on their chemical content. That's the big takeaway.
Crystal clear from the data.
But was there any lingering connection at all? Any faint echo of the old system in the new chemical realities?
There was one small but statistically significant finding, a little nuance. Remember cluster 3, the one with high terpenoline and myrcene and slightly higher CBG.
Yeah.
That cluster had twice as many products labeled Sativa and only half as many labeled Indica as you'd expect by random chance.
Interesting. So that specific chemical profile might have historically been associated more with effects people called sativa.
It's possible. It suggests there might be some historical kernels of truth or association linked to specific, less common chemical profiles, even if the broad categories themselves are scientifically inaccurate for prediction.
But it's a subtle effect, not a rule.
Fascinating nuance. Ultimately, though, the paper's conclusion is pretty clear. It's in public interest to devise a classification system and naming conventions that reflect the true chemotaxonomic diversity of this plant.
Absolutely. We need a new language. So consumer-facing labeling should be grounded in chemistry, maybe listing dominant cannabinoids and terpenes, guiding consumers to products with reliably different sensory and psychoactive attributes.
That's the goal, making cannabis a truly compelling case study for how we handle complex plant-based medicines in the modern world. Regulation, understanding, communication.
Right. What does this whole situation tell us about broader challenges in things like herbalism, pharmacognosy, the study of plant medicines, and even integrative medicine?
It exposes gaps, doesn't it? Gaps in regulation, in standardized naming, nomenclature, and in public understanding based on science versus folklore. The study, despite limitations like not having genotype data for every sample or potential differences between labs in different states, provides a vital basic guide for cannabis product segmentation and classification rooted in product chemistry.
It's a foundational step.
A really important one, yeah, towards a more scientifically informed future for all plant-based therapies, potentially.
Okay, so let's try and recap everything we've dug into today. It's been a lot.
It has.
First, the long-held myth of indica sativa predicting effects. Largely busted by the chemical analysis, the labels don't reliably match the contents.
Check. Reality check delivered.
Second, chemotypes classifying based on actual cannabinoid and terpene profiles offer a far more scientific, reliable way forward.
Definitely a more promising path.
Third, the entourage effect remains a compelling idea, but it needs much more rigorous, controlled research to move from hypothesis to proven fact.
Yeah, the jury's still out, scientifically speaking. Needs good human trials.
Fourth, existing legal definitions, like the 0.3% THC limit for hemp, are strikingly out of sync with the actual science, creating real challenges for growers and CBD producers.
A major policy headache rooted in outdated definitions.
And finally, there's immense, largely untapped potential for innovation in breeding entirely new cannabis varieties with unique chemical profiles beyond just high THC.
Huge potential waiting to be unlocked. We've only scratched the surface.
Which leads to a really provocative final thought building on that last point. If the full chemical landscape of cannabis has yet to be truly explored and defined, what entirely new therapeutic or maybe even recreational experiences might still be hidden within this ancient plant, just waiting for science to unlock them?
Unknown unknowns.
Exactly. And how might we, as consumers, as a society, best prepare for that future? A future where cannabis products are perhaps truly understood, predictable, and maybe even optimized.
That's a deep question to ponder. What else is in there? What combinations haven't we even conceived of yet?
It's exciting and maybe a little daunting. So as you, our listeners, consider that, think about this practical question too. Based on everything we've talked about today, what information would you most want to see on a cannabis product label now? Forget indica sativa, what would actually help you?
Dominant terpenes. Cannabinoid percentages beyond just THC-CBD. Ratios.
Maybe. And how might this deep dive influence your own choices next time, or even just the conversations you have with others about cannabis? Something to think about.